thought I'd get yours.
Are you being facetious or being serious?
Yeah.
No, we missed the blood lecture. So I'm going to do blood and kind of combine white blood cells with lab, and then we're going to do blood typing. Stacey says blood typing, but blood typing is, I'm not going to try to get through all of it today, at least not the first, both times.
We're gonna
It's a little difficult. And I apologize, I only have half of your exams graded. My husband flew into San Francisco, and I didn't realize he was coming up for business, so we met in Fairfield for dinner last night.
That's neat.
I chose that for the grading your exam, so I'm sorry.
Inexcusable going to see your husband.
We choose romantic over
I know, but
working.
if I didn't do that, I wasn't going to see him for 6 weeks, so that's the longest we've not seen each other, so. You know what, we're actually like.
Fight for love.
So me, myself, who's in a contest with my daughter to, if we lose the same amount of weight by the end of the March, she said we could take a cruise and she would pay for it. So we ended up at a Louisiana soul food house. And I'm a vegetarian to boot. So there's collard greens, which I love, but with turkey. And I did have some deep fried okra. And macaroni and cheese looked to die for, but I didn't have any of that. My husband had deep fried chicken and
Everything's deep.
so
I need to go to
I
that
was good
place.
though. I had salad, Caesar salad, and I didn't even find the dressing until I only had 3 bites left. It was stuck under the bowl. And then I did share his banana pudding with him. So
Ooh, banana pudding.
I was like, well, I can't believe we're at this restaurant, but anyway.
Yeah.
So we're looking at blood. Anybody remember from Bio 430 what type of tissue blood belongs to?
Connective.
We've been reading ahead. I didn't expect the answer to be that fast.
No.
All right, so how did I describe connective tissue as cells within a, anybody remember that phrase? Cells
bucket.
within a
Matrix.
matrix.
Oh,
All right. So bone had a hard matrix, cartilage had a semi-solid matrix, fibrous connective tissue had a fluid but lots of fibers in it. So blood fits that description, the matrix being the plasma. So as we look at the characteristics of blood, one of the labs that we no longer do because it's body fluids was to, and maybe you're happy if you don't like pain, was each student got to stab their finger
Ooh, no.
I would look forward to that.
No.
or stab somebody else's. You'd be
Okay.
surprised. You know,
Don't
there are students that asked me to stab their fingers for them because they couldn't do it themselves. And you would draw it up in a tiny capillary tube,
Oh, wow.
and not that big, and plug up one end with clay and put it in a centrifuge and spin it. And when you spun it, I mean, this tube, we're talking, this is just a collection tube, but you're gonna have a whole bunch of blood tests. But it would be a capillary tube. You have a diagram like this on the lab for today. Put a bunch of clay at the bottom because if you spun it, obviously everything that's the centrifuge, if you go around a corner, you go against the side, you go around the outside of the corner. And so in a centrifuge, for about 3 minutes, the heavier molecules and cells get pulled to the bottom. And so you end up with a small glass tube filled with blood that's dark red on the bottom and a clear yellow at the top. So that's similar to what's shown on the tube diagram of your lecture notes, except just larger. And then you can figure out the ratio of blood cells by volume to blood cells by length. And again, this is what you're going to be doing for lab. So you measure the overall length of the tube that contains blood in milliliters. And you measure the total length that are blood cells. Now, most of these are red blood cells, but at the top, there's a small white layer known as the buffy coat. And those are the white blood cells and platelets. So let's say that this is A, and this distance is B. You take a percentage. So the length in millimeters of B divided by the total length in A will give you a percentile. And so that's what is seen in this larger image. And that value is known as a hematocrit. the answer comes out in a percent. For women, it's just under 50%. For men, it's just over 50%, right? So for women, and you'll see a variety of ranges, but typically about 38 to 42%. You might see 40 to 45. And for men, it's about 48 to 52. And again, you might see as high as 50 to 55%. So what we're saying is that's the volume of total blood that's taken up by the cells. So another term that's used instead of just cells is formed elements. The red blood cells, or erythrocytes, are obviously cells, but in the process of leaving the bone marrow, they have lost their nucleus. It gets extruded and pushed out. So they're not a whole cell, all right? So these are red blood cells minus their nucleus and almost everything else in the cell, because they're basically sacks of hemoglobin. Okay, they do have mitochondria and so on, but not much of the organelles. Leukocytes, which are another name for white blood cells, and these are cells. And then platelets, which are really sacks pinched off of large cells found still in the bone marrow. So these two components here are really not true actual cells. Platelets even less than erythrocytes. So formed elements becomes a more accurate term. The fluid that is left is identified as plasma. Okay, that's our matrix. You may have heard of the term serum. Does anybody know the difference between plasma and serum? So if you get a vaccination for flu, or you get bitten by a snake and you're given anti-serum, it's serum with antibodies in it. All right, so we don't want it to clot when we're storing it. So plasma, if we subtract the clotting proteins, we have serum. Plasma is where we have blood functions more than just, we think of blood as, okay, you need blood to transport oxygen and amino acids and glucose and other nutrients. But we also transport hormones, okay? We transport iron, we transport lipids, LDL and so on. So all of those become soluble in the plasma. So if you look at the top of your lecture notes, which I would normally have this on a PowerPoint slide, I list a bunch of different functions for blood. And almost all of these functions have nothing to do with the cells. All right? The transport of gases. First of all, most of our carbon dioxide is transported dissolved in the plasma. A small amount is transported on the hemoglobin, but most of it's transported in the plasma, either dissolved or as carbonic acid. Oxygen is the single reason why we need red blood cells. You would not be able to transport enough oxygen dissolved in your blood if you had no red blood cells. Okay, so you need the red blood cells to carry enough oxygen to stay alive. So that's the red blood cells. The white blood cells, I don't even have on here, and that's defense. Well, we all know white blood cells are there for defense. Everything else, transport of nutrients, hormones, transport of waste, fluid and electrolyte regulation, and temperature regulation, is all the plasma functions. So let's look at some of the major proteins that are found in the plasma. The first one listed is albumin. I'm still on the first page, down near the bottom. So this is, notice I spelled it with an I, not with an E. So this is not the egg white protein, all right? But this is an osmotic control protein. So it helps to regulate osmosis. This becomes very important when we look at the lymphatic function of this unit. Remember we talked about edema, congestive heart failure, right-sided congestive heart failure, blood backed up into the vena cava, and we get systemic edema. Where is that fluid leaving the cardiovascular system? What blood vessel type? Capillaries. So normally, it's normal for fluid to leave the capillaries because of the hydrostatic pressure. What allows even less fluid that normally crosses over is the presence of albumin. And I'll repeat this tomorrow when we cover immune organs. But this is the arterial end of our blood system, and this is the venous end. We have higher blood pressure closest to the heart, and lower blood pressure, hydrostatic pressure, the farther away we move from the heart, so that makes sense. And as we move through the capillary beds, because they're just a simple squamous epithelium, hydrostatic pressure is going to cause fluid loss across the capillary walls. What helps to prevent that are large proteins such as albumin. Albumin is not going to be able to cross the capillary wall. And so it functions as an osmotic pressure. Water, if too much water moves out, then it's at higher concentration outside. And so this has an effect to keep water within the capillary because it won't move. Now, as we move towards the venous end and we lose water, we're farther away from the heart, so hydrostatic pressure has dropped. And since we have lost some water already, what's happened to the concentration of our proteins?
It's gone up.
They're gone up. So, not because we have more proteins, but because of the higher concentration. And so we tend, because of the change in osmotic pressure, we actually return a small amount of water, some of that water, back to the venous side of the capillaries. There's still, however, some fluid left, all right? And that's what brings us to the immune system, because we have to get that fluid back to the cardiovascular system. However, albumin is this major protein that prevents too much fluid from leaving. If you have liver disease of some type, one of the signs is a swollen abdomen because of excess fluid. How is that related to liver disease?
The portal system gets backed up?
Well, albumin is produced, not quite albumin, that's part of it, but albumin is produced by the liver. If the liver can't produce albumin because it's damaged, then osmotic pressure is less and more fluid, increased by the portal pressure, more fluid leaves the abdominal wall capillaries in the peritoneum. You've seen images of children, starving kids, stick-like arms and legs but swollen bellies. Not enough protein in their diet for amino acids to be synthesized to make albumin. So the skinny arms and legs for the lack of muscle protein, but the distended belly because of the fluid, unable to be retained in the cardiovascular system. So that's what we mean by an osmotic protein. All right. Can I erase this part right here so I have more room to write the proteins? Yes.
So you mean that if you increase hydrostatic pressure, then fluid leaves more, and if you decrease osmotic pressure, then
Fluid leaves more.
okay. What about if you increase?
So on the opposite side, so this is going to result in fluid leaving. Osmotic pressure is going to keep fluid from leaving or increase its returning back to the vessels.
Okay. And then albumin functions to prevent.
This is going to increase osmotic pressure.
Okay.
Okay? So then another type of proteins are globulins. And there are a variety of globulins. We're going to talk about immunoglobulins, all right, towards the end of this unit. Those are antibodies. So globulins can function as antibodies, but we also have them just functioning to transport molecules such as iron, hormones, that are not going to be transported on their own. So a variety of components will be transported by globules. And clotting factors. So in the plasma exists a series of dissolved proteins that, when triggered, are affected by enzymes, and over a series of enzymatic events result in the formation of long polymerized fibers. So the clotting proteins are typically mostly enzymes, but some of them are the dissolved fibers. So back to my favorite biological material, Jell-O. You don't have the sugar in the Jell-O, you just look at a gelatin packet. You add hot water to it. As it cools, it firms up because the powdered form of the protein is polymerized to long strands and traps water, so water doesn't flow, and you have a semi-solid. Okay. So many of the clotting factors are enzymes that trigger, but we also have dissolved proteins that, triggered by the enzymes, will polymerize and trap the blood and keep it from flowing, and that's a clot. All right. So both clotting factors and clotting proteins are found. The clotting factors are proteins are found in the blood, and then we have other enzymes and hormones. When I first finished my master's and moved back to Tennessee, one of the senior biology students that I was at the college I was teaching at was doing a project, and he was wanting me to help him put an IV into a rabbit, and he was going to replace the rabbit's blood with a chemical fake blood that would bind oxygen. And then you could put the animal in underwater, not really water, but in a solution of this same type of chemical, it looked like milk, and it would have such a high concentration of oxygen that it would be like a fish. They could get the oxygen from this fluid and transport it in its blood and not need to breathe air. So there have been a lot of different types of research. This was back in the early 80s, 83. And there's been a lot of type of research, especially for war zones, when they don't have enough plasma and packed blood cells and stuff to provide to individuals. So you need both of these components. You need the fluid and you need the blood cells. There was a case in California where a woman was having liposuction done. She'd had it done previously, and she was going in again. Her husband dropped her off at the clinic. It was an in-and-out clinic. And he called a few hours later, and they said, well, she's not ready yet. So he called an hour later, and they said, well, she's still not ready yet. It was like 4 hours past the time that she should have been done. So he drove over there, and she had died. And what had happened was, obviously the people involved were trained. They just kind of, we can do this. They hadn't gone to, he was a medical doctor, but hadn't had any special training in it. And they used different types of equipment, but this was the early where they would have a cannula, which is a tube that's slanted at one end. It has a cutting edge, and it's hollow, so it's attached to a vacuum, basically. They slide it under the skin, and the cutting edge, they go back and forth, and slice up the adipose tissue into small pieces, and the vacuum sucks it up. Well, in doing so, they had cut some significant blood vessels since they didn't know where they were and what they were doing. And so they gave her blood pressure was dropping, so they gave her IV fluids. Well, it got into a cycle where as the more she bled, the more her blood pressure dropped, the more IV fluids they gave her, which they should have been giving her blood, because IV would help to keep the blood pressure up, but, so it's the volume, all right, but it's lacking blood cells. So eventually, and clotting factors. So they're diluting out her blood cells and diluting out her clotting factors, and eventually her blood was so dilute that she just continued to bleed because nothing was clotting and she didn't have the oxygen transport. So they made the mistake of not being able to admit, we're in over our heads, we need to send her to the hospital, an emergency room, because they didn't have the blood supply on hand to do that. They just kept adding water, essentially. Even though it had glucose and electrolytes, it didn't have all the other rest of the material that blood has. that she needed, primarily the clotting factors and the red blood cells. All right. So now let's look at red blood cells. So our word for red blood cell is erythrocyte, cyte meaning cell, and erythro referring to red. All right, so what color is blood when it's deoxygenated?
Dark red.
Thank you. My kids said, swear, when they were in elementary school, that it's blue. And it's like, well, our teacher said it's blue, Mom. When you look at your hand, your veins are blue. And I said, well, you guys have had your blood drawn. It's drawn. They said, well, it becomes red as soon as it hits oxygen. That's why it looks red when you cut yourself, because it hits oxygen in the air and it turns red. I was like, well, honey, you had your blood drawn from your, for blood tests, and it goes into a vacuum. There's no oxygen in the vacuum tube. It's not blue when it goes into the vacuum tube. But Mom, our teacher said. That's what I'm the mom and not the teacher. That doesn't work how much I argue. So there are some individuals that have methemoglobin, and yes, then they're still not quite blue, but their skin looks a little bit more blue. This is the most common number, volume so about 4.5 to 6 million. And again, you'll see a variation on these numbers per cubic millimeter. So remember those tiny little squares on the ECGs? That's a millimeter. Okay, so that size cubed is what we mean by cubic millimeter. And these develop in the bone marrow, stem cells. You guys remember where red bone marrow is found? So in the sponge, red bone marrow is where we find spongy bone. In the fetus, it's found in almost all the bones, all right? So it's found in flat bones and long bones, both the epiphysis and the diaphysis, where we have the yellow marrow that we talked about in Bio 430. And in addition to that, it's found in the liver and in the spleen. Okay. In the adult, it's limited to flat bones and the ends of long bones. The epiphyses. So the most common flat bones from which red bone marrow is obtained is the ilium. They usually go into the iliac crest. They need to do a quick sample, say in the emergency room. They'll usually go into the sternum. Interestingly, one of the most, remember when I was in the coroner's office last week, one of the most different things, it was very similar to a embalmed cadaver, but when we were cutting through the sternum to open up the rib cage, Jeff was doing it, and big garden clippers to cut through the sternum. And red bone marrow just kind of oozed out. And I've never seen, it without being solid, more like a solid tissue. So, I don't know if you've ever seen fresh liver, but if you take that and put it in the blender, it was kind of that consistency. But a dark red, not a brown. And I've always just thought of it more as bony tissue rather than the the more fluid tissue that the red bone marrow is. So I was teaching at the med school, residents that were working in the emergency room would come to use our cadavers at the end of the quarter and practice doing thermal taps to obtain that bone marrow. So stem cells are always there, all right? The earliest stem cells look the same for both white blood cells and red blood cells. And they're not in color, but I have an image of them on page 5. And you don't need to, I'm not going to show you slides of these various types of cells and have you identify them. It's one of the most difficult labs that the medical students have to do is identify the various stages of the stem cells. Even for us as faculty, but what made it nice was their quizzes. Every Friday, they had the microscopes, and we would quiz them on stuff. But it was up to them. The quiz would say, put the arrow on an unequivocal example of da-da-da-da-da-da, whatever we were studying. So for red bone marrow, it would be, put the arrow in the microscope on an unequivocal example of a hemocytoblast or an erythroblast. And so I couldn't tell what it was, then it was an unequivocal example. So it had to be something that was so characteristic that, because even for me, I could not identify every single cell in the microscope because there's a continuum and some of them are kind of in between these particular characteristics. But what I want to clarify, what I want you to understand is the processes, what they go through. So what events are occurring. You look at that first cell versus the last cell. So we start out with the hemocytoblast. And then we go through various erythroblasts. So hemocytoblast, there's nothing in that term that says red, right? It just says blood. Hemo refers to blood. And so there aren't any characteristics. It's already been determined in its DNA whether it's going to be a white blood cell or a red blood cell, but we can't tell at this particular stage. And then it goes into various stages of erythroblast. So what did I tell you was the primary content of a red blood cell? Hemoglobin. So the whole purpose of this cell's developing is to synthesize hemoglobin, right? So you'll notice that these first cells have nuclei. So a nucleus is present. What are the two main purposes for having a nucleus?
Ribosome.
Messenger RNA, protein synthesis. And what else?
Meiosis.
Mitosis, cell division. So these early erythroblasts can undergo mitosis and produce additional ones, and they are synthesizing messenger RNA primarily, obviously for the enzymes needed for this, but primarily for hemoglobin synthesis. Once sufficient messenger RNA has been produced, the nucleus is no longer needed. Because the messenger RNA can be utilized over and over on multiple ribosomes to continue the amino acid sequencing to produce the enzymes and the hemoglobin. So notice what's happening to the nucleus. You get smaller and smaller and smaller. What's happening to the chromatin as the nucleus is getting smaller? More and more and more tightly coiled, right? The more tightly coiled the chromatin is, it's not going to be copied for messenger RNA synthesis. So once we're done with our messenger RNA synthesis, that chromatin material becomes more and more and more tightly coiled until we have a very small, the term pycnotic is used. Let's see, I don't think that first C is there. I think this is how it's spelled. It goes from this to very tiny, 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 and highly compact. And then it's finally extruded from the cell. It leaves the cell. At the same time, the cell is synthesizing hemoglobin, all right? And it loses ribosomes, and so it becomes pale, because the ribosomes make the cell look bluish purple. And so as it loses those ribosomes, it becomes more and more pale. And so it finally enters the bone marrow as an erythrocyte without the nucleus, without ribosomes, and basically a sac of hemoglobin. However, if there is some form of anemia, or there's some drive to make additional red blood cells, and they are entering at a faster rate, they're being produced at a faster rate, and they enter into the blood with still ribosomes present, or ribosomal ER present, then they are known as reticulocytes. And it's these little dark little strands that you can see in the cell. And that's a sign, the more reticulocytes that are present, it's a sign that the body is under stress to produce additional red blood cells. Okay. So leukapheresis is under there for formation of new white blood cells, and we'll come back to that when we talk about white blood cells. So now let's look at some characteristics of red blood cells. Anybody remember the diameter of a red blood cell? When we talked about capillaries, I mentioned this.
What's the diameter?
Approximately 7 microns. So from here to here is approximately 7 microns. So it's a good gauge. Almost every tissue has red blood cells in it. So when you're looking in the microscope, you can see the red blood cells and estimate the size of other structures or cells around it. You'll notice its shape is a dumbbell shape when we have it cut in cross section. And this is identified as biconcave. It takes some really interesting feats of bioengineering that were studied in the 80s and early 90s to understand how the proteins were linked together to keep it in this particular shape where it's like a ball that's been pitched in the middle. What do you think is the advantage of having a shape like this?
It's squeezing.
If I have a sphere,
It pushes more to the outside.
you're close. So if I have, remember the purpose of this is to transport oxygen. So if I have oxygen bound to hemoglobin, it's almost equidistant from the surface anywhere within the red blood cell. Maybe not equidistant, but it doesn't have too far to travel. Where if I had a sphere and I had oxygen in here, that would have quite a considerable distance to diffuse into or diffuse out of the cell. So it makes it easier to pick up and bind and easier to release oxygen to the tissues. Within the red blood cell is a molecule known as hemoglobin, and I'm not going to try to diagram that picture on the board. Hemoglobin is 4 strands of proteins, 2 alpha and 2 beta. And within each molecule of hemoglobin, there are 4 iron atoms. Is oxygen electronegative or electropositive?
What? Negative?
Electronegative. Iron ions are able to give off electrons, and so that's the attraction between the oxygen and the iron. Okay? So an oxygen molecule will bind to each iron ion. Now, when we get to our respiratory system, We will talk about, we will talk about the attraction. So it's more difficult for the first oxygen to combine, and then it becomes easier after that. And it's more difficult for the first oxygen to come off. I mean, it's easier for the first oxygen to come off, and it gets more difficult. So we'll talk about that relationship when we get to the respiratory system. On the next page, on the bottom of the next page, is a illustration of the life cycle. Red blood cells don't have a nucleus, so they have a limited lifespan. Once they're released into the circulating plasma, their lifespan is approximately 120 days. It's about 4 months. Their membrane becomes unstable. They have a tendency to collapse and rupture when they are forced to conform their shape too much. And so they are then recycled and broken down, and primarily in the liver and the spleen. So those organs that were sources of new red blood cells in the fetus are sources of recycling in the adult. So you can see on the diagram, which I would normally have had up on the screen, they're produced in the red bone marrow, and they exit the red bone marrow through wide capillaries with larger in diameter than our normal capillaries, with gaps between the epithelia. And this is a site where we have sinusoids. And so it's easy for these large cells to pass out of the capillary when normally even, the LDL proteins, covered proteins have difficult, protein-covered lipids have a difficult time. So they circulate in the blood, and then they are broken down by macrophages, which are a type of white blood cell in the liver and the spleen. We'll talk about, they have a special name in the liver called the Kupffer cells, but we'll get to that when we get to the digestive system. And then amino acids. Content of the hemoglobin is recycled. And the iron is recycled. There is a pigment known as bilirubin that is excreted primarily in the digestive tract as part of the bile that the liver releases. That's what gives the bile and fecal matter a kind of a yellowish brown color. Now, if the liver is breaking down too many red blood cells, okay, or the liver isn't able to process the bilirubin, then the bilirubin can build up in the blood and enter the skin and results in a yellowing of the skin known as jaundice. So this is due to increased bilirubin concentrations. It's typically not a problem in newborns unless it rises, let me rephrase that. It's usually the rate at which the bilirubin increases, it's more of a problem than the actual total amount. When babies and moms used to stay in the hospital for 3 days after birth, these numbers have come down. So when my older sister was born, my mom had to sign a release after 10 days to leave the hospital. They wanted her to stay longer. It was a British clinic in Burma. And she swore that if she didn't finish her food, they brought her the same plate back again the next meal. So my dad put a little mark under the plate, and sure enough, that was the same. Well, I don't know if they just used the same plate over and over, but it was the same food or not. But she had to sign herself out after 10 days to be able to go home. Then it was 3 days, standard when I was working labor and delivery. And then when my kids were born, it was overnight. So less than 24 hours. Well, when it was 3 days, we would do a bilirubin test in the nursery, and that was when I was working in the nursery. If it wasn't labor and delivery, that was my job, and we had a little machine in the nursery itself, and I'd do a heel stick and run the bilirubin, so we had a standard before the infant left for home. So why do you think an infant gets newborn jaundice? Why were they breaking down so many red blood cells? Anybody have any idea? Remember we talked about the fact that the baby's blood goes to the placenta and picks up oxygen across the capillaries from the mother's blood. It doesn't get the mother's blood, but the oxygen transfers. And so the fetal hemoglobin, again we'll talk about the respiratory system, has a higher chemical attraction for oxygen. It's different, the alpha chains are different. And so once the baby's born, adult hemoglobin is needed, not fetal hemoglobin. And so the fetal red blood cells are broken down. And so there's a period of time where there's a big breakdown and recycling of fetal red blood cells. Also, if the baby is held below, with the umbilical cord still attached, if the baby is held below the level of the mother's heart, additional blood can enter the baby from the umbilical cord and placenta that normally would stay in the placenta. So that overload of additional blood cells can lead to increased jaundice. And so if the bilirubin rises too fast, that can affect the brain function. So that's why there's an issue with that. It was found almost serendipitously by some nuns that were working in a nursery. They noticed that babies whose bassinets were closer to the window didn't get jaundice as much as babies that were closer, deeper into the room. And so it was discovered that ultraviolet light, the energy of ultraviolet light, helps to break down the bilirubin that's in the skin. So when my twins were born, I was teaching 5 classes at 4 schools, but a lot of them were in the evening. And so I was home Tuesday and Thursday mornings, and I got home after 10 on Monday and Wednesday and Fridays for a few hours, and so my mom was out to help with the kids. And so I would take one baby, go outside. They were born in September, so it was still warm. Spend 10 minutes outside, which is the baby in his or her diaper, and then go back in and bring the other baby out, because that ultraviolet light helps with that chemical breakdown. If a baby has high bilirubin and has to be hospitalized, they'll put it in what's called a Bili incubator, and they'll put dark glasses, basically gauze, over its eyes so that the ultraviolet light doesn't affect that, and basically roast it, not really, but turn it from side to side so the skin is exposed to the Bili light, the ultraviolet, kind of a purple light that you'll see these babies in. And that helps to keep the levels down. But it is normal for babies to have a little elevated bilirubin for a period of up to 2 weeks after they're born. Okay, let's look at some blood tests, and then we'll talk about some types of anemia. And then for white blood cells, I'm going to have you go ahead and start the lab when you're looking in the microscope for these cells, because that will really, rather than actually lecture on them, because that will increase your recognition of their characteristics. So blood tests, we've already discussed hematocrit. So I want you to be able to recognize for the lab exam, even though we're not doing an actual hematocrit, recognize the type of equipment that would be used. So if on a lab quiz or a lab exam I show you this equipment, you would be able to write down the name of the test. Okay. So the centrifuge and is shown on the right. You see a tabletop centrifuge. It doesn't have to be the big ultra centrifuges that are used in research. And on the left is the little capillary tube with clay. So after the blood is drawn, it's stuck into that clay. Hemoglobin, there's a variety of ways of testing hemoglobin. That's a machine that reads wavelength, color wavelength. But when I was, we were still working with body fluid, we would take a piece of blotting paper, and after we had filled the capillary tube with blood, we would dab our finger on the blotting paper. And this was way long before you worried about exposure to other people's bloods. There would be a card that had printed on it various shades of orange to red. And there would be a hole in the middle, and then you would just lay that over your blood and read what the color was. So if you were reading, so if this was, you just slid this back and forth until the color of your blood, that was still red, matched the color on the card. That'll have various shades of orange to red, but that's what you would see here. So this would be the lower hemoglobin value. And this would be a higher hemoglobin value, be a brighter red. So you go orange to dark red to bright red and match up the color. So hemoglobin is given in grams. I believe it's grams per deciliter. And for females, it's about 12 to 14 grams, and again, I believe it's per deciliter. For males, it's more in the range of 14 to 16. So when you're pregnant, they usually test your hemoglobin. I don't remember if it's 20 weeks or 28 weeks. When I was pregnant with the twins, mine came in at 8. And I just thought I was tired because I was carrying twins. So they said you can either start taking iron pills, which nobody likes because they increase constipation. So I said I'd rather eat greens, so I would eat a bunch of collard or kale greens every day, the entire bunch. And that helps take care of the iron problem. But of course, iron is utilized in red blood cells. So if you have low iron, you have low ability to transport oxygen. Without enough oxygen, you have decreased ability to make ATP in the mitochondria. So that's the consideration there with that aspect. And we'll come back to hemoglobin synthesis when we look at anemias. Sedimentation rate. This, I don't know why we even still talk about it, but anybody given blood lately? Did they do a sedimentation rate when you gave blood?
What is it?
They'll take your blood and they'll pull it in a tube, and then they measure the rate at which it settles.
Yep. Oh, they did it different the other day when I gave it at the blood drive. They put it into a little, it was like a flat thing, and they put it into a machine.
Okay, that would have read the hemoglobin. Sedimentation rate is so vague. If you look at the physio activity, it can indicate an inflammation. It can indicate certain, but it's so vague, it's just, there's something wrong if you have a high ESR. What basically happens is red blood cells, if there is an increase in certain proteins, if there's an inflammatory process, the red blood cells will start to stack on top of each other. Kind of like a stack of pennies. Well, they're all the same size. I have them getting smaller and smaller here, but that's not. And this is called a rouleau. And this is increased, so as this increases, it results in an increased ESR. And there are certain diseases that cause a decreased ESR. And it's also affected by gender and by age. So it really just is an indication that some other tests have to be done. Okay? But so the more rouleau that's created, this is heavier than a single red blood cell. So it's going to settle faster. And it's done at a rate, over how many millimeters over a time period. So if you look at the image on page 10, these are different samples, and you can see that they're settling at different rates. But it's a really vague test. It doesn't indicate anything specific.
So when I went to give blood, they did something like that. They said because my blood dropped slower, I was anemic, and that I could not give blood at that time. So
That's what they test. They usually
Yeah.
test the iron. They usually have to do mine like chronically.
Yeah.
But it's definitely hemoglobin is a much better way of testing for anemia. This isn't a direct test for anemia. It's more of an inflammatory, it's not the best test exactly for anemia. Because anemia has to do with a couple of different things. In fact, that leads us to our next page. Anemia. So anemia can be due to iron content, It can be due to protein content, so actually the numbers of red blood cells, or the amount of iron available. So you can have different issues with anemia. But essentially, any anemia will show up as a decrease in hematocrit and a decrease in hemoglobin. So, one cause is hemorrhage. Having made the state that anemia will show up as a so what you'll have is, this could be due to menstruation, could be a cause of hemorrhage. You may not think of it as that, but, and a whole week is just losing a cup, not all of that is blood. But if you have a very long period, all right, or a heavy period, or comes more often than once a month, then that's going to increase the likelihood that you're going to have blood loss. There is hemolytic. The most common cause of hemolytic is malaria.
Is that just destruction of red blood
Yeah,
cells?
so it's the rupture. More rare would be a electrolyte disorder, but that's really serious because you're going to lose a whole bunch of other cells, not just red blood cells. All right, so it's the red blood cell rupturing, and in malaria is because of the parasite life cycle that's causing it to rupture. And if you have a lot of rupture, this is what leads to a very serious form of malaria called black water fever. And the reason the blood is black water is because of all the hemoglobin that's being dumped out as the red blood cells rupture. Aplastic. This is usually an unknown cause. It's a disorder of the bone marrow. So it's usually it could be radiation, but often they don't know it could be a chemical exposure. All right, so it's some problem that's causing the stem cells to not produce more red blood cells. And again, it's usually a work-related environmental toxicity kind of cause.
If they're not producing red blood cells, then how are they treated?
If it's really serious, they have to have a bone marrow transplant.
Okay.
They have to replace those stem cells. Iron deficiency is what it says. So that's what I had. Not enough iron in the diet. So I might have had the protein, all right, but the red blood cells and hemoglobin didn't have enough iron to transport oxygen.
Is there a tie between jaundice as a baby to iron deficiency or any of these others?
Not that I know of.
I'm just curious, 'cause I had jaundice as a baby, and then now every time I'm iron deficient, so I just...
I'm anemic. Every time, I haven't even tried to give blood probably in 15 years because every time I went, they don't accept it, so I gave up.
Did you have jaundice as a baby?
I don't
He's a
remember my mom saying
vegetarian.
anything about it.
You're a vegetarian, so it's...
So is the rest of my family, and they aren't anemic.
It could be, because there's a difference between heme iron and non-heme iron.
Yeah.
You could just not like non-heme iron.
My husband does eat meat now. His blood type is O negative, they call him a lot to give blood. Pernicious anemia is one of the more interesting. This is what I should have if I'm a vegetarian, but I'm not a true lacto-ovo. Pernicious anemia has to do with a lack of vitamin B12, which is used in the synthesis of red blood cells. So you can buy vitamin B12 in drugstores or grocery stores. You can get injections of vitamin B12. People do that to get more energy, get more red blood cells, get more oxygen. So vitamin B12 requires a molecule produced by the cells lining the stomach known as intrinsic factor for absorption. So even if you have it in your diet, you have the vitamin B12 in your diet, if you don't have intrinsic factor, you're not going to be able to absorb the vitamin B12. Another factor about vitamin B12 is it is found only in animal products, right? So I do not eat fish, white meat or red meat, seafood, but I do have milk, all right, and cottage cheese and yogurt, and those are animal product components and have vitamin B12. If I wasn't a true vegan, then I would need to take vitamin B12 as a pill. So it's a little difficult to be a true vegan because that vitamin B12 actually comes from animals too. This is processed. So now, if you lack intrinsic factor, taking vitamin B12 as a pill isn't going to work. Because it doesn't matter if you take it orally as a pill or if you take it orally in food, if you don't have the intrinsic factor, you're not going to absorb it. So if you lack the intrinsic factor, then you need to take the vitamin B12 as an intramuscular injection and bypass the whole absorption across the digestive tract.
Where does the intrinsic factor produce that?
The stomach lining?
Oh, okay. 'Cause
No.
I have to get this shot.
Okay,
I had
yeah.
gastric bypass surgery, so I didn't.
Yeah.
They never told me why.
In fact, that will be a question on the digestive system exam. Person has gastric bypass surgery and had also their spleen removed, and so which of the following is a problem? And with the lack of the stomach tissue, they lack sufficient intrinsic factor.
They don't tell you, just.
So can you be tested for intrinsic factor? I mean, is that just, or do they just make some?
I don't know how, I'm sure they can, but I don't know how they would do that, if it's a lavage, a stomach wash, or if it's, they just, I'm a DNA kind of thing, I'm not sure. Good question, I'm not sure how
Do
they
you
test for
have to
it.
get the shots?
Whoa.
Oh, be careful.
You
Do
what?
you have to get the shots?
No, because I eat animal products.
Oh, okay.
So because I have milk and products and stuff like that, cheese, then that has vitamin B12 in it.
Yeah, but if you don't have the intrinsic factor,
Oh, if you don't have the intrinsic factor, then you would eat shots.
yeah, even if you eat animal products.
Correct. Because you would have, if you eat the products or if you took the pill, you wouldn't be able to absorb it. So the intramuscular injection is, you do it once a month or, okay. And that bypasses the requirement. This has nothing really, it just involves vitamin B12 as a side story. But this was the first case where, since we're going into the immune system, where testing the sequences, knowing the sequences of the HIV virus was used in a forensic case. And I've told some of you about this story. There was a woman who was married with kids, having an affair with a guy who was married. I don't know if he had kids or not. He was a medical doctor, and their ongoing affair was for several years, and they both said they were in love with the other person. And, well, let's change partners, divorce, and then we'll marry. So she did. She divorced her husband. He never divorced his wife. So after a few more years of continuing to see him without the benefit of marriage, she got tired of this undercover relationship, so to speak, and said, forget you, I am moving on, and started dating other guys. Well, he kept interfering in these relationships, and she had some kind of dependent relationship. She would get dragged back into a relationship with him. She finally found somebody that wanted to marry her, that she loved, and they were developing a good relationship. And so she told the guy, this is it, this is over, I don't want you coming by anymore. Well, a couple of days later, he called her up really late at night, and he said, I'm going to come over and bring you a vitamin B12 injection. Now, some guy, some ex-boyfriend called me up and said they were going to come over and inject me. I would have just said no. So this shows the degree to which they had that codependence. It was like after midnight. And he had done this before when they were dating or seeing each other. And so he came over and gave her a shot. 6 months later, she developed HIV. And she was not a nurse. She didn't have any surgeries. She didn't have any risk factors or groups that she belonged to. And she was convinced that that was what he had given her in the supposedly vitamin B12 injection. She went to the local police, and they were like, you don't have any proof. You know, he's got a good reputation in the community. We're not just going to go disrupt this. Surprisingly, they listened to her after repeated visits, and whatever proof she had, that she had no other source of it, and they were like, well, it could have just been contaminated, whatever. And she said, no, this is our history. You know, he was getting back at me for dumping him, so they got a search warrant. And they went to his office. I'm not sure what the setup was, but the end result was they were able to find blood from a patient of his, still stored in either the lab or some refrigerator, on-site or off-site, from a patient that he had seen the day that he had gone to her house. And the patient had HIV, and the labs were able to sequence the RNA in the HIV virus, and it matched. And because viruses mutate so rapidly, that was unusual, unless it was from the same source. But not only that, the patient also had hepatitis C. And she had that virus and the same sequence as well, so it was even added proof. She ended up dying from HIV within 8 years after that, and he was convicted of murder. And it was the first time that using that type of fingerprinting of a viral was used in a forensic case. Okay, sickle cell anemia. Then we'll take a break and do some lab. Sickle cell anemia is a genetic mutation, a single amino acid substitution, and I believe it's, well, the two amino acids are glutamate and valine. And this results in a destabilization of the membrane such that when oxygen concentration in the blood, not what's bound to the hemoglobin, but what's soluble, so it's called oxygen tension or oxygen pressure, the CO2, when that drops, the individual goes into oxygen crisis, or sickle, not oxygen crisis, but sickle cell crisis. And normally round cell collapses to a crescent shape. This is not stable, it becomes stickier, it tends to clog up in the capillaries, especially in joint areas and in the organs, and so it can lead to very painful joints and damaged organs because of the lack of oxygen. As I said, it's genetic, so you can have the trait. It's genetic, you can have the trait and not have the disease, or if both parents have the trait, then the child can have the disease. It's primarily seen in African Americans that have a more direct African, what's the right one? Ethnicity. So it developed in Africa, and so, I mean, if you've been in Africa, if you've been in the United States for, 200 years, you're not as likely, that population is not as likely to have sickle cell as someone who's just immigrated, a population that's just immigrated from Africa. Interestingly, sickle cell provides a protection against malaria. Okay, so people who have, the other way around, malaria provides a protection, no, they're right the first time. Sickle cell provides a protection against malaria. So if you have sickle cell, the malaria life cycle doesn't complete itself within the red blood cell. And so there's a kind of a, symbiotic is not a true term for it, but there's a protection there that's included with that. Okay, the last word on the list before we take a break here is polycythemia, and that's just the opposite of anemia. So with polycythemia, you can have, this is less common, it's too many red blood cells. So what's that going to do to the viscosity of the blood? It's going to increase it. So typically, the polycythemia is due less to abnormal red blood cell synthesis than it is due to other causes, so dehydration or other forms of changing the viscosity. So that would have a high hematocrit. So my husband lives in Denver, which is at 5,000, well, Mile High City, so 5,280 feet. It's some parts of it it's hilly, so it's not all exactly 5,280 feet. When I was there at Christmas time, we went down to Colorado Springs to a hotel which was at 6,000 feet. So at higher elevations, you're going to have less oxygen molecules for every 500 mils that you breathe. And the body responds to that by increasing red blood cell synthesis. Ever heard of EPO?
Yeah.
It's a bad word in the sports world, at least for if you're trying to compete. EPO is a hormone produced by the kidneys. So when there is less oxygen, such as high altitude, it will stimulate hematopoiesis, hence its name, erythropoietin. So some athletes will inject themselves or with EPO to stimulate increased red blood cell synthesis so they have more oxygen transport and more energy for whatever athletic event they're doing. Without EPO, there's blood doping, in which blood is drawn, spun down, the blood cells, so packed blood cells are stored, and the plasma is removed. And then just before an athletic event, return to your body. So you're not getting a transfusion from somebody else, you're getting your own blood cells back. So obviously that's going to increase hematocrit. So again, these are kind of artificial, unusual forms of increasing red blood cells, but it would result in polycythemia. So in the lab, let's take a look at your lab. What I want you to do now is to take a look, we're going to, after break, we'll look at white blood cells with the microscope, and that'll give you a better practice at the characteristics. But if you take a look at the assignment on page 11 in the lab packet. So this first assignment here, these white rectangles are representing capillary tubes. So they all have the same amount at the top. So your total length is going to be the same for all of them. That's what the black bar at the top is representing. And then the light black line is the shaded area should be a little bit darker than that. But that's the formed element. So the formed element in number 1 is going to come all the way up to here. The formed element in number 2 will be to there. The formed element in 3 to there. And the formed element in 4 to there. So I'm asking you to figure out the hematocrit. So you would just measure the distance for each of the formed element volumes, length divided by the total length of the tube. And that total length is going to be the same. So remember, total length is from here to here. So measure that to the dark bar, the black bar at the top. So that's your total length from the bottom to the black bar, the bottom of the black bar. And then you're going to divide that number into the length of the formed element for each of those, for the light line. So like I drew on the board earlier. So this represents 1, this is 1, and this represents 1 sample. So you would take this length and divide it by this length. The other way around. Take B and divide it by A.
Formed element on
And
top.
that will give you your percentile of whatever the volume of formed element is. So that's what you're going to do for each of the hematocrits. And then the bottom is not the days of the week. These weren't drawn on Monday and Friday. Those are genders, so those are male and female individuals. And so for a male, is that hematocrit abnormal? For a female, depending on your patient, is that abnormal? And then I want a cause. So, look at the bottom. I do not want anemia or polycythemia as the cause. That's the condition. So we've talked about various causes. Obviously, you don't know for sure, but I just want a potential cause. And I don't want the same one. So if you have 2 cases of anemia, I don't want you putting iron deficiency for both of them, okay? So give me a different distinctive cause that might result if you have polycythemia or if you have anemia. What is some potential cause that could be? Okay? All right. The other factors of processes are from PhysioX. And so we're going to come back and start and talk about platelet plug formation and blood typing. But for now, I want you to take a break, and then we're going to take a look at white blood cells under the microscope. Okay, I'm going to see if I can get the computer work so I can show you some pictures. But your lecture notes basically go through descriptions and functions of each of these. So I'm going to save time there by not repeating all of that stuff on the board. But I would like to show you images of them, and then you'll get to find them in the microscope. Okay? So, 15 minutes. With is the rarest cell. So everybody tries to find it. And that's a basophil. So basophils have granules. They typically are less than 1% to 2%. So a couple of you may find some. They're a little bit larger than lymphocytes. Okay, so look at the size of the cell relationship to the red blood cell. And then look at the lymphocyte in relationship. So they're similar in size, but the basophil is slightly larger. Now, look how coarse the surface of it is. That's the way you're going to distinguish between what you think is a lymphocyte or what you think is a basophil. It might really be a lymphocyte. So basophils have granules. What you can't see is the fact that their nucleus looks like a horseshoe. But the granules are so big and so dark that they obliterate the nucleus. So it's hard to see. But if you look closely at the surface, those granules will make, see how irregular this is? It's not smooth, like the edge of the nucleus. So when you confuse this with a lymphocyte, you're confusing the dark nucleus of the lymphocyte with the granules of the basophil. If you look for that coarse surface, you don't see a clear cytoplasm rim like you see with the lymphocyte. You want to look along the edge here, and the granules make this an irregular surface. So that's the key to figuring out whether you're looking at a lymphocyte versus a basophil. Okay? These are also involved in, they're associated with IgE, but not as a specific. So these cells will bump into IgE after they've already bound specifically to pollen or to toxins, and will release histamine. So they're part of an allergic reaction. The other two are a little easier to identify. So the eosinophil is the fourth as far as finding it. So this is going to be around more than 1%, and it ranges anywhere from 2 to 3%. Some places will just leave the basophil at less than 1, all right, and then the eosinophil is just above that. It also has a horseshoe-shaped nucleus, but it's acidic leveling, that's why its name is eosinophil, and it has distinctive red to orange granules, pink or salmon color. The nucleus is still visible. These granules are slightly smaller. And the key thing about eosinophils is they increase in parasitic infections. So if you go camping and get Giardia, or if you come back from a foreign visit with worms, or your kid's playing in the sand pile and gets worms from eating sand that had fecal matter in it, all right, you're going to see an increase in parasitic infections. I mean, an increase in eosinophils. So these are pretty distinctive. If you look over here on the right side, okay, you can see these are the eosinophils here. I have a slide of eosinophilia, so there's a ton of eosinophils in the disease pathology slides over there. Finally, the third most common is a monocyte. It also does not demonstrate granules. And this is a very large cell. Okay? This is going to be about 8 to 10% of the population, and again, it varies. This becomes a cell that you know well. This will become a macrophage. So as a monocyte, it's not functional. It has to convert to a macrophage. It's the largest of all the cells. In fact, it will not be nice and round. It will be collapsed by other red blood cells and white blood cells around it. Its nucleus kind of looks like it's folded over on itself or kidney shaped. So practice looking at these cells for the next 35 minutes or so. 30 minutes, at least until you feel comfortable, and we'll come back to lecture and talk about blood clotting. Okay? So I'd like to do that about 10:15 or so.
Sure.
No, I can only look at one eye. Take his emergency.
Clear. Well, to me, it still looks like a whole bunch of bumps.
Yeah. Can I twist it? Ooh, pretty. So the one that,
Do we use a ruler for
Let
the?
me grab some.
Oh, for that first.
Yeah.
Okay.
But I'm
on the left is
Oh yeah, we are. I was
The
just.
one on the right, Olympus side. We can probably focusing more.
I know. That's what I need. I need help. I moved it to.
looks
What
like
is that
the
on?
thing
10?
is folded over kind of.
So I started with the low one. I focused. I started with 10. I focused it.
On the one on the right.
I mean, I semi-focused it, but it looks the same as when it was at the 40. So now this is.
all over the cell, and it looks like this is
You can move the thing as well.
Oh, it's kind of awkward for me from this angle, but no, I definitely see the sickle cell.
Oh yeah, they're all.
Over by the arrow?
I see what looks like.
The one on the right, do you think it's a monocyte? Because it has that smooth edge.
when you are
There's one of the longest ones.
Yeah, the monocyte on
allowed
the
to
right.
sure and clean off both your
No,
judges
she said it
and
was
the slides with
bigger
the oil with
and
with
isn't
extra.
round because it's much higher there.
Are those the only 4 things cells?
Yeah, those are kind of the 5 things that you find in blood.
Here, let me try to find it on the lower one.
You can tell the difference.
Can we ask her? You want to ask her? We can ask her for it. It doesn't look, because it looks like the inside is folded over, so you think that, they think it's a lymphocyte? I think it's a lymphocyte.
The blue one? Big blue one? Yeah. Yeah, they're kind of smooth.
And if you find something you think is a basophil, raise your hand.
Get one of ours and see if you think it's a
Yeah, let's
lymphocyte.
and then I'll come.
Looks like that's an eosinophil. That's right in the center.
Nope, that's a neutrophil. Off the top.
Or neutrophil.
And then
The one on the
One at the bottom is a
the one
neutrophil.
that's like dead center.
This one up here at the
I think it's
top. Yeah, so
or neutrophil, I mean.
Well, it does only have two.
that one's a neutrophil.
I'm looking at it because it's granular and it's red.
So just that only dark purple thing in there, that's
Yeah,
it.
so see how the nucleus is kind of multiform?
Okay. Because we have a slide with iron deficiency.
Oh, okay.
Getting anything?
Yeah, but I don't know. It looks like a
Folded nucleus, not a folded nucleus to me though.
It's kind of out of focus when you're looking at it.
What do you think? Can you see like any particular cell?
I think so, but it's out of focus. But everything else around it is just right.
See, that's what I keep seeing, and then I can't see anything. I think we're supposed to be able to do more than that. Then again, we haven't done the right oil. How do we do the oil stuff? No, did you guys do the oil stuff already?
Yeah.
Okay, so maybe that's what we're having. You have to do the oil stuff, right? You can't.
7 microns.
This one looks like a plasmon. It kind of looks like a basophil.
Yeah, it looks like two totally separate things, so I think you're right. I think it's a neutrophil.
They're on that side.
On the right.
Hold on just a second.
It's not, it's definitely not
I
big.
think we have to.
Right, which is why I think I lost it. Hold
So the one on the right, yeah, the one on the
That is an eosinophil.
Which one?
So I'll clean that
The
one.
one on the, very pale though. Oh, well. It's,
Is it the one that's got the red granules in it and the
yeah,
purple?
but the nucleus is all broken up.
So I thought it was a neutrophil because of
The nucleus does look like a neutrophil, but the granules are too large. But let me check some other, nope, you're right, it's a neutrophil. All the other ones look like that. So, yeah, the granules are just more distinctive.
They were supposed to do
So,
I know.
yeah, but no, the granules are
Just that we
that.
have to do oil emergency.
Here is a collapsed monocyte. Turn, you can turn.
Do you think that that one was a monocyte next to nearby with the?
Okay, this one's collapsed, so it doesn't show, I didn't see that one. So this one doesn't show much cytoplasm. But monocyte nuclei, another way that you can tell monocyte is the nucleus has wide, white, almost like clear areas in it. Like lipid droplets, but they're not lipid. So you'll see this kind of the monocyte. So take a look at that. That's a monocyte there. Doesn't show much cytoplasm because it's, but let me ask you that question.
How do you do the oil immersion?
Go between,
Yeah.
I've never done it before.
green and the white lines. So right halfway between, drop the oil on and then go to the red.
Yeah, see Amy, you put it in between two so it's open.
Would have been a monocyte.
And then a drop of oil.
Yeah, so they're trying to identify every.
Because you don't want the oil to touch the objective, right? You do on the oil lens. So this one is, you're just going to slide it. Oops, I'm moving the table.
You want us to put it down?
Stage? No. It just slides in like that.
Really?
These are definitely.
So this is the only one that's oil immersion one? Oh, yeah.
I put my hand on the table, so I might have really like moved it. Oh, look it! I found it.
One that she said had.
Do you guys have yours on the 100 one?
Yes.
No, ours is on the
It's on the, yeah, 100. That's the oil.
Is that not the?
No, that's.
Crap, that's the wrong one.
That's the
That's the wrong one? You want me to lower it?
Our beds. That's what I was wondering. I was like, which one's the.
Is that done all the way?
You guys didn't take micro yet?
No.
You use the microscope a lot.
Really? Okay. So you get really our focusing is horrible. At least my
We
focusing is
have
horrible.
a monocyte.
Can I see, please?
Are you not supposed to read about that one?
It's hard to get it, it's harder to get the oil out.
Once it, when you turn
The
it,
lens is kinda
it
hidden.
changes the focus because it changes the depth of the.
I totally lost what I was looking at when I did it.
Yeah.
You'll just have to, when you change slides and get to that one, if it's kind of blurry and you can't focus it, there's the oil on it and just keep hitting it.
Yeah, there's some that are really small.
The monocyte is the one you're pointing at.
The main thing.
It's the blue one. There's only one blue thing in there.
You can go like until it's
Very long.
Yeah.
The purple thing, the
Yep.
big one, okay. Got the discussion about the purple the other day.
And then we should be able to focus.
Purple, and then.
It's gonna take a while to figure out where to focus it.
Yeah, it's really dark.
If it's dark, you can
Shape though.
go that way.
Oh, it's bright.
Betting is.
So that's a
Oh, I thought you
monocyte?
were calling me over, I don't know.
Yeah. Cause I'm
That's a collapsed monocyte. It has all that, the blue over to the side, so it's just seeing part of the cell.
I see what you're doing. It's not complete. It's not wrong.
To the blood typing. If you've had micro, you've probably already discussed blood typing. If you haven't, we'll be looking at it and figuring out. It ends up being more difficult than it needs to be. Because if you take it slowly and look at the principles, it should not be confusing. But if you allow it to start off confusing, it tends to stay that way, okay? So that's why I want to do this now and kind of go through it slowly, and then we'll see if we have time for our, the lab for identifying for blood typing is a fake lab, well, the blood is fake. Because again, we don't, that was a fun thing, is for everybody to figure out their own blood types. And we just haven't gotten back to that point yet. I mean, it's sterile techniques and all of that, but we'll see. My only reason not my only reason, but one of the major reasons why I don't fight more against our labs using, testing your own, because you can set it up where you're only testing your own blood, you're, doing all of that, you're not working in pairs or anything, you're just testing your own. But about 15 years ago, a student of mine was a diabetic, and he had gotten a new glucose monitor, and he came in and he said, here, look at this, why don't you try out my new glucose monitor? So I said, okay, and I set it up, and I opened up a new Lancet and put it in there and poked my finger and tested it. And then he tested it, and after he tested his, I said, you didn't change the Lancet, did you? And he was like, his face was pale, and he was like, you don't have anything, do you? And that was just the two of us in the room, and as often as he had done this, he'd only done it for himself, and so it just never entered his mind to change the Lancet. And that little kind of mistake like that can be devastating if somebody has HIV or hepatitis or something like that. So that in the back of my mind has made me less willing to push forward and say, I really want to do blood labs, because any little mistake, even though you need to learn how to be sterile and careful and stuff like that, it's just a little mistake could be devastating. There was a professor who's retired now, when he was teaching at Sac City, did a blood typing lab. And they were looking at, if your parents are O and A, and what would your possible blood type be. And so he came up with his blood type and had asked his parents what their blood types were, and he brought it to the professor, and the professor said, that's not possible. He said, what do you mean? He said, this is my blood type, and this is what my parents told me their blood type was. He goes, well, then they can't be your parents genetically for you to have this blood type.
Yeah.
And he was a college student and had never been told he was adopted.
Oh,
And so he went home to have this conversation with his parents, and they were furious with the professor. But that was what had come out in the lab, kind of a thing. So most of you have probably heard of blood types. Sorry about that.
I want to find out more.
Yep. Start from day one, let them know. My niece knows.
What?
My niece knows she's adopted.
What blood type are you? A positive? I'm O positive.
I think I'm A positive.
Oh, yeah, see,
I know I'm A, I don't know if I'm positive or negative. I'm pretty sure I'm positive. Get it?
oh, like you did not take off the hard hat.
Sorry, that was a lame joke.
It's okay.
It's confusing, it gets confusing.
It has
All right, so there are several different blood types, and we are looking what cell are the markers, what we call antigens, are the markers present on when we determine blood type.
Say that again.
What cells have the markers or antigens from which we determine blood type?
A and B.
Red blood cells. Okay, so these are these surface markers, surface proteins that are present on a red blood cell, which I'm going to draw in black because that's the only thing I can find. And they are distinctive formats or shapes that represent the three antigens, which are I mean, in microbiology, I don't know if you looked at more than the general blood types, but there are smaller variations, many small other variations that occur than the dominant ones that we're going to talk about here. But there are A antigens, B antigens, and D antigens. The D antigen is also known as the rhesus factor, which yes, is a monkey. And I'll talk about how that name came, or it's also known as the terms positive or negative are used depending on whether you have or don't have that D antigen. All right. So this is just showing the ABO component. So here the circles are representing the A antigen, the triangles the B. So if you have the A antigen, you're considered to have blood type A. If you have the B antigen, you're considered to have blood type B. If you have both antigens, your blood type is AB. And if you have neither A nor B, your blood type is O. Okay, so that's how we identify the ABO component. So the blood type you have identifies the surface antigen on your red blood cell. Now, in addition to that, then we have the D antigen. So if you have A and the D antigen, your blood type is considered, we'll form a table for this, your blood type is considered to be A positive. If you have A antigen and you don't have the D antigen, your blood type is A negative. All right? So that component is really, really clear and simple. So let's focus on that first before we start muddying up the water. So blood type and antigen. If blood type is A, and we won't look at the positive or negative yet, then you have the antigen A. If blood type is B, you have the antigen B. If blood type is AB, you have antigen A and B. And if blood type is O, you have neither A nor B. Okay? And I'm going to come back to the D antigen again in just a moment. Now, my daughter's just telling me she got 100% on her paper. But she stayed up till 40, right? So sorry about that. All right, so it was one of those group projects where two kids don't do anything.
Oh, I hate that.
You've all had those, haven't you? So when we have, the next thing we're going to look at now are the antibodies. Now we haven't talked about antigen antibodies yet, and we'll do that next week. But typically, anybody get the flu vaccine this year? Okay, did you get sick? Mm-hmm. Yes. 80% of the people I talked to that had got the vaccine got the flu. But anyway, so vaccines are a weakened virus that is injected into you to expose your body to the viral proteins so that your B lymphocytes produce antibodies against them. And if you get an actual true viral infection of strong viruses, you have these B cells that have the memory of the antibody synthesis process already sequenced, and they can rapidly produce antibodies. Antibodies are of different forms. We'll talk about IgG, IgG, and E and M and so on. But typically they have this Y shape. They may be dimers or pentamers, but they typically have this Y shape. Whether it's a G or an E or an M type antibodies dependent on this region. The Y ends, the forked ends are specific to the shape of the marker that they're recognizing. So what happens when we have a blood cell that has the antigen on it, the antibody that would recognize that is capable of binding to not just that antigen, but the same antigen on an additional cell because of its forked Y form, and now these two have been clumped together, okay, or agglutinated. We'll go through those terms. So you don't want to give somebody blood that they are going to have antibodies against, because so if I am type B and I have antibodies against A, my antibodies will clump the red blood cells and they end up usually rupturing as a result of that. So if you have antigen A, you're going to have in your blood the antibodies against B. So you're going to have the antibodies against the antigen which you don't have. Doesn't make sense to have antibodies against the antigen you already have, right? Now, these antibodies are, have you ever heard of eating for your blood type?
No.
I have a friend who was trying to lose weight and got on that, and it just never made sense. It was something that started in Japan, and it was like, what does your blood type have to do with what you eat? I mean, that's one of those. I've read that if you have migraine headaches and you have too much potassium on the left side of your body, or too much magnesium on the right side of your body, it's like, this is the bloodstream. It mixes all over the place, you know, so how could that be? And my daughter said, well, Mom, I read that if you have hormonal acne, then you'll have a breakout on the right side of your face if your right ovary is the one that's ovulating. It's like, well, honey, the hormones all go into unless it's getting there through the connective tissue, it all goes into the blood. So eating for your blood type never really made sense to me until I started researching last year, actually. All right, why do these antibodies come? Because normally you need some exposure to the antigen. You don't just develop antibodies. You have to have exposure to the antigen to stimulate the B lymphocytes to make antibodies. But babies are often born, it seems, with the antibody to the antigen they do not have. So I thought, this isn't making sense on the typical mechanism by which antibodies are born. So I talked to Lori, Dr. Smith, and I did some research. And we knew that the fetus, the embryo, doesn't develop the antibodies. It is based on the food that they eat. It's actually antibodies against antigens on the food. So antigens similar to A that are present on foods that you eat, you're not going to make antibodies against because your body has already been desensitized to forming antibodies against its own blood type. But if you eat foods with B-type antigens or exposed to those in the embryo from your mother, then you would make antibodies against those because you don't have the B antigen. So now eating for your blood type, I still don't understand it, but it does make a little bit more sense than it did before. So you're not just automatically, your B lymphocytes don't just automatically make the antibodies if you have the A antigen. It is dependent on exposure during the fetal time and shortly after birth to the foods that you eat. The end result is still the same, however. So if you have antigen A on your blood type, on your red blood cells, in the plasma, you would have circulating the B antibody. Okay? if you have on your red blood cells the B antigen, so if this is the A antigen, this would be the B antibody. Sorry, the other way around. That would be the A antibody, but you would have in your own plasma the B antibody circulating around. All right? So this would be B antibody in your blood if you had A antigens on your red blood cells. If you had B antigens on your red blood cells, then circulating around instead, so if these were B, you're going to have antibodies against the A antigen. Everybody with me so far? Okay. Now, if you have both antigens on your blood type, you have A and B, it really wouldn't work to have any antibodies, right? Because then you'd be agglutinating your own red blood cells. So if you have both A and B antigens, then you don't have antibodies against either A or B. And if you do not have A and you do not have B, then you're going to have antibodies against both A and B. Okay? Yes?
Do you know of a hypothesis about why the body does this? Why does the body do this? Do you have,
I have no idea.
Like the gut scales.
yeah, this doesn't make sense to me why you would have antibodies against another kind of
Well, our typical cells, all of your cells have self-markers, okay, that are specific to you. What I don't understand is the A and the B are not specific to you, because we can't just transplant the liver from one person to another. Those markers need to match as closely as possible, and even still, you have to take immunosuppressive drugs. But we can transfuse blood types if they're the same. So I don't know why they're not body specific. Because all of our cells that have nuclei have those body specific components.
I just wonder if it's to protect us.
So I don't
from
know if
something.
it's a modification of that process. Red blood cells don't have nuclei, they've lost them, so, but no, I don't know why they have this particular aspect to it. All right, so now, yes?
So you would need antibodies that would work like that are opposite with the antigen. Unless
You don't want to
have
have antibodies that are going to clump your own blood.
Okay.
So you're not going to make antibodies against yourself. Okay? I mean, sometimes we do. That's what rheumatoid arthritis is, and diabetes type 1 are considered to be autoimmune diseases. But you're not born that way. You don't develop that from embryonic development. All right, so this is the easiest part to understand, so I want to make sure it's clear because this is essential to the next step, all right, which is who do you donate to and who do you receive from. So, you do not, let me draw a picture here. So if you are the recipient, this is your blood, okay? This is recipient blood. And this is donor blood. So this is what we're adding. Now, you do not want to add any antigen to blood that has antibodies against it. Because what will happen is that agglutination, and you'll have transfusion disorder, high fever. It can cause the agglutinated bloods to rupture. Okay, and you'll have a transfusion reaction. So, if I have red blood cells that have A antigen on them, my antibody is going to be what? My antibody is going to be B. So what will happen is if I add a red blood cell that has the B antigen, this antibody will agglutinate it. Okay? So you don't want to think of the antibodies as sharks swimming around in the pool. You don't want to jump into that pool if you're the food that the shark eats. So you don't ever want to add the antigen to the antibody. Okay? So the next thing we're going to do then is look at the antibody. If there is an antibody present in the blood, we don't want to receive, we're the recipient, so we don't want to receive any blood type that has that antigen. All right, because our blood has the sharks. This is the shark. All right. So we don't want to put into the pool anything that the shark is going to eat. This is what the shark is going to eat. The antigen, not the A. So the B antibody is going to eat B antigens. Sorry for the anthropomorphic term, but sometimes telling a story helps. Okay? So, unfortunately, I need to move over here. So, if this now can receive from what blood type?
You know.
So we want anything that doesn't have the B antigen. So it can receive from A. What other blood type doesn't have the B antigen? O. All right, so we can receive from A and O. But did you see the process, the thought process by which we figured that out? I don't want you to have to memorize this table, okay? Should be able to figure it out. All right, if we have the A antibody, we can receive blood that has anything that doesn't have the A antigen. So we can definitely receive from ourselves. So B and O. All right. Now, type AB blood doesn't have any antibodies.
Nope.
So it can receive from A and B, AB, and O. Does that make sense how we figured that out? Okay? Type O blood has both A antibodies and B antibodies. So we can't give it anything that has the A antigen or the B antigen.
Just
Oh.
O then?
Just O. Okay? Before I move on again, if you look at that process, all right, that you cannot give the antigen to the antibody, then you can't give whatever blood type you have. So if I say, okay, blood type O, what type of blood can you receive? The next question should be, what are my antigens?
There's none.
Blood type O has no antigens, so what antibodies do I have? A and B. Therefore, I can't give blood type with A and B antigens. So you see the sequence of thought processes there? Okay, so you can figure it out without having to have that whole column memorized. Now, the next question is, who can I donate to? It's a bit of the reverse. You can, it's not the ideal situation, all right? But you can donate antibodies. They get diluted out. What you can't donate are the same thing. You can't donate the antigens. So let's look at an example. So my antigen is A. I'm the food, remember? So I cannot give my blood type to anything that has sharks that will eat me. So I cannot give A to anything that has A antibody. So we're doing it in the reverse. So now I am, to draw that picture again, I am the blood coming from the beaker, going into the pool, and this blood cell has A antigen. Therefore, I cannot give it to any blood that has the A antibody, because it will agglutinate all of my blood cells. So we're just reversing our thinking process I am now the food, and I'm not going to jump into a pool that's going to eat me. On this one, we were the pool, and we could not give ourselves anything that the sharks would destroy. So, if my blood type is A, and I'm donating it to somebody, they cannot have the A antibody, because they will agglutinate me.
Okay, so it has to be.
So here we have the A antibodies. So can I give myself to myself?
Yeah. Because you don't.
So I could give A to A, because A doesn't have antibodies against A. Can I give A to B? No. Because it has antibodies against A. Can I give A to AB?
Yes.
Yes, because there are no antibodies.
And not to O.
Can I give blood to O?
No.
No,
They
because they have both. See how we figured that out? Same principle, just looking at it from a different angle. Okay, now I'm blood type B. Yes.
The AB. The AB shouldn't it be?
Oh,
Oh, yeah.
no, I can give to AB. What are you asking?
I thought we were making a list of who A could donate to,
Yeah, A can donate to A.
and also to
She's
AB.
saying you should put the AB right after A.
Oh, thank you. Yes. Okay, so now blood type B can donate to B and can donate to AB for the same reason, but it cannot donate to O because there's antibodies against the B, all right? Now AB, can AB donate to A?
No.
Wait, AB? No.
No.
Nope, because there's antibodies against the B part of my AB. Can AB donate to B?
No.
No, because there's antibodies against the A part.
So it can donate to yourself.
It can donate to itself. Can it donate to O?
No.
No, because that would be a double whammy. There's antibodies against both. Okay?
Oh, so O can't give. Can O give to something? No, it can't.
Oh, I'm sorry. Put it in the wrong place again.
Oh,
All right, O.
it can give
to O.
So O doesn't have any antigens.
So it's
Oh,
going to
so-
everybody.
But what antibodies, so yeah.
But it
So
has. Oh,
who can it donate to?
it has A.
Everybody.
Everybody.
No. It can
It
only
doesn't have
donate
AB
to
antibody?
AB.
You can donate to AB as well?
Anybody.
So
donate to
it has no antigen, so can it donate to A?
Yes.
Yes.
Because the antibodies in A, and the B antibodies in A have nothing to bind to.
Oh.
Can it donate to B?
Yes.
The A antibodies have nothing to bind to. So also it can donate to AB,
And then to
and it can donate to O.
So, okay.
So, isn't it universal, we call it?
We're not quite ready for the universal, I mean, but yeah, the universal donor, but a certain, not just O. All right?
So when you look at the donate to, you have to look at the antigen part.
You have to look at the antibodies. So if I'm donating, my antigen is what? So let's say my antigen is B. So I have to know what my antigen is first. And then I have to look at their antibodies to make sure they don't have antibodies against me.
So, O is
Okay.
like, in your analogy to the sharks, it's like, oh, you're just not even shark food.
It's
You're an enigma. All right, so practice this. Again, don't memorize the table, but practice doing that, figuring it out. All right, so back again before we move on. So this column, if I'm receiving from, remember the most important thing is what the antibodies are for in the person that is getting the blood. So here, what are the antibodies in the person I'm donating to? Here, what are my antibodies? Okay, so if I'm receiving, the concern is my antibodies. If I'm donating, the concern is the other person's antibodies. Does that make sense? Because that's the bigger pool that's going to destroy whatever's coming in. So if I'm receiving, I want to look what my antibodies are, and I can't receive anybody that has antigens that match my antibodies. If I am donating, what are my antigens, and then the person that I'm donating to can't have any antibodies that match my antigens. Okay? Now, plus and negative. Babies were being born that had a disease known as erythroblastosis fetalis. Not fetalis, fetalis.
In your notes, it's an E.
And what happens is, I think it's an A.
Fetal like fatal or fetal like
Fetal. Fetal life. Trying to get myself enough sleep so I can do this. What happens is in the mother, if the mother is of a negative blood type, doesn't have the D antigen, when she is exposed to that D antigen, she will develop, or anybody, will develop antibodies against it. The difference with the A and the B is they're exposed to it early, apparently, from food. All right, either from the mother or food that you eat early on, but usually from the mother, because you're born with these ABO and these AB antibodies, so food from the mother that crosses the placenta type components. Now, if you are, let's say that you're O negative, and you're given O positive blood. The O positive blood has the D antigen. Your B lymphocytes will respond to that, recognize it, some of them will, and you will develop antibodies against the D antigen. Normally you are not born with those. So if you are never exposed to, if you're O negative and you're never exposed to O positive blood, you will never develop those antibodies. All right, so that requires exposure. The concern with erythroblastosis fetalis is that the exposure comes from the baby. So usually at birth, when the placenta separates, and there's tearing of the chorionic villi and so on, the baby's blood can be mixed with the mother's blood. And after the baby is born and safely out of the uterus, if the mother is negative and the baby is positive, meaning the father had to be positive, then the mother will develop antibodies against the baby's positive antigen, the D antigen. That baby is already born. It's going to be fine. The next baby, however, that is positive, those antibodies can cross the placenta, and while the baby is still in utero, will cause agglutination of the baby's blood cells. It can result in death before the baby's even born. So back in the 40s, 50s, 60s, these babies were born very ill or dead, and they stillborn. And so they started doing some research and discovered this additional antigen. Well, first they did the ABOs. Okay, they found those, but then those weren't sufficient. So they used the rhesus monkeys, and they used prisoners from a prison in Nashville that volunteered early release time credits for working in and they found the D antigen, or the rhesus factor, because of the rhesus monkey component. And they at first treated them, after the baby was born, they would give the baby transfusions. Then they were still getting babies that were born that were dying or were already dead. So they started doing transfusions in utero. That still wasn't saving every baby. So now the procedure is to give any woman who is negative, who has a negative blood type and is pregnant, an injection during the pregnancy. They used to do this about within 3 days after the baby was born. If the baby was positive, the mother would get a RhoGAM shot. And next week we'll talk about what's actually in that. It destroys the baby's blood cells before she has a chance to mount an immune response. But they were still losing some babies, so now whether her baby's positive or not, the mother gets this injection during the pregnancy. I think it's twice now. And so in case there are any circulating fetal blood cells, the mother doesn't have a chance to mount an immune response. So although we'll go into that more detail next week, or maybe tomorrow, that's how the discovery of the D antigen came to be. So any of these blood types may or may not have the D antigen. If the blood type is a positive, then the D antigen is present. Okay? If the blood type is A negative, then only the A antigen is present, and there is no D antigen. If the B positive, we have B and D. B negative, we have only B. AB positive, A, B, and D. AB negative, A and B. O positive, just D. And O negative, nothing. Okay? So again, a little more basic concept. Now, antibodies. If you have the D antigen, you will never make the D antibody. Just like you don't have antibodies against A if you have the A antigen. Does that make sense? Because you'd be a good in yourself. If you don't have the D antigen, you will only make the antibody if exposed. All right, so you're not born with it. Exposure to food doesn't result in it. So you have to have some form of a transfusion, or if you're a mother, having a baby that is positive. So you'll only have the antibody, the D antibody, if exposed. Okay? And again, the D antibody if exposed. D antibody if exposed. So if my husband is O negative, he obviously hasn't carried a child. So unless he's had O positive transfusion, he should not have an antibody against D. He would only have antibodies against A and B. All right? Here. So that need to prevent, we never want to create antibodies. So we don't want to give positive blood to negative because that would trigger the antibody. And if it got positive blood by mistake, again, he would have a transfusion reaction. All right? So, now, let's go and look from the received from. If I am A positive, I definitely can receive from both positive, I can receive positive blood, because that's what I already have. The negative means I don't have the D antigen. There's nothing wrong with not getting something. So I can also receive negative. Does that make sense? So positive blood can receive positive and negative. Positive because I'm already positive, so I can't do any damage to myself. Negative has nothing in it. It's like receiving A blood with no B. The B is absent, so there's nothing wrong with that. So I can get positive or negative if I'm positive. If I am negative, I can only get negative. Because giving myself positive would result in making the antibodies. All right, so I can, if I'm A negative, I should only receive A negative and O negative. Does that make sense? Because I want to prevent that immune response. If I'm B positive, I can receive positive and negative, positive and negative. Again, there's nothing wrong with getting the negative if I already have the antigen. There's nothing wrong with getting blood without the antigen. If I'm B negative, I can only receive B negative and O negative, because otherwise I would start to make antibodies against the D antigen. If I'm AB positive, what could I get?
Everything.
Everything. So this is known as the universal recipient. blood to have if you're selfish. Because in danger or emergency, you can receive anything. AB negative can only receive the negative form of each of these. If you're O positive, you can receive O positive or O negative. And if you're O negative, you're out of luck. You can only get O negative.
Okay.
Which is why my husband is, so O positive is the most common blood type. So I used to wonder, why are we always getting these phone calls? Why are people always calling me to ask me to donate blood? It's like, well, duh, if it's the most common, it's what everybody needs, because most of the people that need blood are going to be O positive as well, okay? And then my husband's O negative, so he can go to either O positive or O negative people.
If you're O negative?
You can only receive O negative,
Oh,
but you can be donated to both.
Oh, okay.
So these people are out of luck. They can only get O negative. All right? Now it's okay to be O positive, even though you can only receive O positive, because so many people in the world are O positive, it's not difficult usually to find some for donation. So now in reverse, the other side of the coin, so to speak, the same thing applies. If you're O positive, you can only, if you're a positive, you can only donate to positive. If you're negative, you can donate to both positive and negative. All right? So A positive can only donate to A positive and AB positive. A negative can donate to both.
Why is that again?
Because they are negative and they have nothing
Oh, no.
to add.
Okay.
Okay? So B positive can only donate to B positive and AB positive because we don't want to add the D antigen to someone who's AB negative. So if we gave B positive to B negative,
We'll be adding.
they would develop antibodies against the D antigen. So B negative can go to either.
Okay.
Now, if I'm AB positive, I can only go to AB positive.
Okay.
If I'm AB negative,
We'll go to both.
I can go to both. Okay? If I am O positive, I can only go to A positive, B positive, AB positive, and O positive. But if I'm O negative, like my husband, I can go to everybody.
Oh, and that's because negative.
So this is the universal donor.
And that's because negative.
AB can also go to A or B.
No.
I can't donate AB to A.
Because it has.
A has B antibodies.
That's true. Right, okay.
So universal donor.
So a drug can only do.
So notice, just a sec, notice that O negative, the poor person with O negative can only receive O negative blood. And yet their blood, they don't get any of the benefits. They're very limited. They can only receive one blood type, but they can give to all the others. What was your comment, Noah?
So it can only give to O negative though, in the end? The O negative blood type?
Oh, sorry,
Or,
I left it off.
and that's because it won't be given no sign, like it won't give any of them the D.
Right.
So the D antibody is only if present if you are positive. And then,
The other way around. You're positive if the D antigen is present.
okay, and then, so you can only give it to other positive blood types because they already have the D antigen.
Right. Okay, now we'll do our lab on this next week. It'll hold because it's artificial. Because I want to do some practice
Did she say how you get the
questions here, and therefore then our blood type will be a little easier. I'm afraid if we do the blood typing lab now, you'll just be confused. If we look at the antibodies in a different way. So, let's say, I'm going to just throw you guys in the pool, so to speak here. We'll start out with the very most difficult question. Well, maybe not the very most. You're looking at somebody, and they have B antibodies in their blood, what would their blood type be? Their blood type is A. Would it be positive or negative?
We don't know.
You don't know. Okay? It could be A positive, which they wouldn't have any D antibodies, or it could be A negative, in which they had never been exposed. And in that case, they still wouldn't have any antibodies. All right? Let's say that this individual has no A or B antibodies, but they have D antibodies.
Oh, they don't have A.
So wouldn't the, on the first question, wouldn't that also be O or no?
No, because what kind of antibodies would O have?
Oh, and.
A and B.
Oh, so it's AB?
So only
it's
A.
AB positive.
Their blood type is not AB.
Oh. There's no A or B.
They have, oh, no A, I'm sorry, I was thinking about somebody's question.
Yeah, I was thinking of the
They
first
have
one.
no A and B antibodies. Let me come back to yours in just a moment. They have no A and B antibodies, so they would be AB. They do have D antibodies. Negative, that's correct. If they're positive, they're not going to have antibodies. Okay, so remember, they don't have antibodies if they have the antigen, and they don't have antibodies unless they've been exposed. Okay?
Okay, so, but if that said D antigens, then it would be.
Yes.
Okay.
That's why I said I was going to jump in the pool and do the hard step first.
Okay.
So no A or B antibodies mean they have A and B antigen. The presence of the D antibody means they can't have the antigen. Therefore, they don't have the antigen and they were exposed to it. Okay? So A and B antibodies only.
Oh, we don't know.
O. And we don't know. So when we're looking at antibodies, the only time we know anything is if they're present and then we know they're negative. If the antibodies aren't present, we don't know. It could be either or. It could be positive or it could be negative without exposure. Does that make sense?
Let's do some more.
Okay? Now, so D antibodies would be positive or negative?
Negative.
Negative.
And there's A and
A antibodies would be the
B.
opposite antigen,
B.
B. Okay? Now, the easiest ones are to look at the antigens, because that tells you what the blood type is. So if we change all of these to antigens, so AB antigen, well, let me just change it. Period. So antigen so if we have A and B antigen only, what would the blood type be?
AB.
AB,
Negative.
negative, because we don't have the D, right? If we have D antigen only, O,
Positive.
positive,
Okay.
we don't have A or B, all right? A and D, A, positive. We're looking at the antigen, not the antibody. So A, positive, okay? So where it gets tricky is if you get mixed up on antigen or antibody.
I know.
So you have to look at that aspect. All right. Now let's say that we have a mother who is A negative, and we have a father who father who is A positive, but he's heterozygous. Or I don't want to write like that. So it's heterozygous for the D antigen. And so they can have a child who is a negative, which way the recessive for the mother and the recessive for the father, okay? And they can also have a child who is A positive. So if the baby is A negative, would there be any problem with erythroblastosis fetalis?
No.
No, because there's no D antigen from the baby to activate antibodies in the mother. If the baby is A positive, and this is their first child, that baby probably will be okay, but will stimulate a response in the mother. So that the second A positive, the next baby is A negative. Any problem? The mother now has D antibodies.
No.
Still no problem, because even if the D antibodies cross into this baby, there's no antigens for it to bind to on the red blood cells. So the next baby is A positive. Now this baby can be affected by those antibodies because it has the D antigen.
So the mother would be
So here's a scenario. The mother has one child who is negative. Okay? Her second child, this one's fine. Her second child is A positive and is stillborn because of erythroblastosis fetalis. Because her first baby was negative, so before they started doing the RhoGAM shots anyway, she didn't get any treatment. And her second baby, her first baby with the D antigen is born, dies of erythroblastosis fetalis. What happened? She didn't have any transfusions.
Oh, she must have had something happened, like in utero, where she had an exchange of some kind when the baby was.
First baby was a fraternal twin, which she didn't realize, and neither did the doctor. And the baby died, say, at 4 weeks, and the tissue was absorbed. At 4 weeks, you have blood cells, and those blood cells had D antigens on them. So even though she wasn't aware, she already had mounted her primary immune response. So this really wasn't her first positive baby, which is why they started doing those RhoGAM shots, because you never know when you might have been pregnant and the pregnancy, you had a miscarriage and you weren't aware of it, or the baby died at a very young embryonic age and was reabsorbed. And those antibodies now are already, the B cells are already primed to mount a second immune response when what you think is your first positive pregnancy or even your first pregnancy. Okay? Let's stop there. I think that's enough mind-blowing.